Cell regulation in the cell. The drastic difference in cell type, even in a single organism, means that there are a wide variety of time frames that the cell takes to go through its cycle. This can be as short as a few minutes to several hours in humans. As these cells are going through their cycle, it is essential that they must be copied exactly in order for the cells to work properly. If a cell isn't copied exactly, then a mutation occurs and will continue to occur and to be passed on until it takes over. If the cell isn't working like it should and passes on this quality, it can cause the body to not work properly. To ensure the cycle continues as it should, there are 3 checkpoints in the cell to ensure the cell duplicates exactly each time. The G1 checkpoint determines if the cell is going to move into the S phase. Before it can do that, it must clear the requirements of the checkpoint that includes favorable conditions. This is where the cell commits to the action of the cell division or not. If the conditions are favorable for that type of cell, then it will divide. If you think of yeast, this is a good example. There are requirements for this yeast to activate, such as it needs sugar and it needs warm water. If these conditions are favorable, then the growth can happen. If the factors aren't being met, then it will either grow slowly or it won't grow at all. The factors determine how fast or slow the cell can grow. If it meets all the requirements, then it will go to the checkpoint. In order to make it past the checkpoint, then the DNA must be undamaged and ready to go on to the next phase. If the checkpoint is not passed, the cell will either try to fix the problem itself, like inside of this process, or it will simply freeze and then just wait for an outside signal to come in. This could be from inside the cell, or it can be from outside the cell coming in to the cell. Once it passes that G1 checkpoint, it moves into the S phase. This S phase is where the DNA strands unwind and the double helix splits. It acts much like a zipper. If you think of it that way, that will help you get a good idea of how it comes undone. This zipper then is bound to the polymers and gains the other half back. So it splits each half, then binds, and then gets the other half back. This is shown in the bottom of that picture. This gives you then 2 identical strands of DNA. This process is kept in check or halted by the P16 protein. This keeps it in check until the DNA is completed and it can move on to the next step. This P16 protein is used in cancer treatments because of its tumor reducing or eliminating qualities. Once the DNA strands are complete, then the cell is ready for the next phase. Very similar to the G1 checkpoint is the G2 checkpoint, where the criteria has to be met before the cell can go on to the next phase. The G2 checkpoint includes things like meeting size requirements and making sure there's enough protein in reserve to make sure that the cell can finish out the process. If these criteria are met, then it moves on to the metaphase. If not, then the chromosomes and DNA don't check out. There must be exact replicas of these in order to go through this check. So it's checking the size and how much protein reserves it has. It's also checking if the DNA is the DNA and the chromosomes are exact replicas, so it can move on to that next phase. If these aren't met, then the cell will either try to complete or repair the DNA until it is cleared on this checkpoint, because since we are in the cycle already, we cannot stop, so it will continue to repair damage or complete DNA until it clears that checkpoint. Once this checkpoint is cleared, it moves on to the metaphase. The metaphase also has a checkpoint. This is the third and final checkpoint. This is where the chromatides that have now been duplicated are attached firmly to the spindle microbules. If these are attached correctly and firmly, then the cell moves on. It's checking to make sure that they are attached and anchored to two fibers from opposite poles of the cell. Until this happens, the cell process will not move forward. Regulators inside of the cell. So this would include those checkpoints that we just went over, and it would include the positive and negative regulators inside of the cell. The positive regulators speed up or simply help continue the process, whereas the negative regulators can slow down or even stop the process if enough of them are damaged. There are two kinds of positive regulators. There are the cyclin and then the cyclin-dependent kinase. And in order to work properly, they must be bound together. So the cyclins have to be bound to the CDKs in order to work. This activates them, and they must be fully activated and phosphorylated in a specific location for their mechanism to work. These molecules are what push the cycle on to the next step. So they have to be present and abundant in order to get that cell through the cycle. So if you'll notice, there is an increase of them at each phase. So when it gets through those three checkpoints, there is an increase in that cyclin in that phase, and then a drastic reduction in it afterwards because it's no longer needed. The opposite of those positive regulators would be the negative regulators, and they have the opposite effect in the cell process. They can either slow it down or halt it. The proteins bind to a site, and then the growth is halted until it is needed again. This is so things aren't growing while other things are getting ready for them. So if you imagine you're building a shelf for your house, and you have your saw and your sander and your finish, whatever you're going to paint it, all together on one table, and it's locked in place and you can't move anything. So your saw is just going up and down when it's on, so you can cut. If the sander's on, then it's just constantly running, and then the paint's just falling, like one of those cool videos where they put it through and it's just finished on the other side. So those negative regulators are on the sander and the finish while you're cutting, and then when you're done cutting, one of the negative regulators attaches to that saw, so it stops cutting the wood and you can go on to sanding it, and then the negative regulator is detached from the sander while you're using it, and then it's attached when you move on, so that only one thing is happening at a time, because you don't want to be cutting the wood while the finish is going on half of it, and then you just have a mess. So you want one thing happening, and then a negative regulator comes in, stops that process, so you can go on to the next one. And then when it's all done, you would simply turn the electricity off, because you don't need them anymore, and so you can move on to the next phase. So then it goes to the next phase, and you've got a whole new set of what you're trying to do. If bear with me for a second. So here is the G1 phase, and then we get to this G1 checkpoint where we check to make sure the cell is big enough and the environment is stable. If we clear it, if you watch that center, it's showing what's happening. We go through our synthesis phase, so now we have our 4. We get to our second checkpoint in the G2 phase. Has the DNA replicated? Is the cell big enough? And is the environment sustainable? We go through our mitosis. You can see the mitosis checkpoint. Are the chromosomes aligned correctly? Are they attached correctly? And then we split, and we are back to our 2 cells. So there it would go again. We looked at one of them, and on to the next one, all the way through that synthesis. It goes from 2 to 4. There's our second checkpoint. Really quick, there's our third one. And now we have 2 cells. And that would be our final piece. Thank you very much. And that